This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 820, recorded on October 21st, 2021. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today here at the incubator in New York, Daniel Griffin.
Hello, everyone.
Daniel's right next to me, which is unusual. We've never done this before. The update started after COVID. So now and then you would come to the Columbia to do a TWiP, but
yeah, there was, right as things were starting, I think it was right at the end of February, early March, I came to the office, right? And I was very, keeping very distant, looking in a different direction.
That's right.
I was already very worried.
That's right. That was maybe, February?
Yeah, we should look back. But yeah, that was the first, right at the end of February, early March, just when it was,
So this is actually not the first time I've seen you, because we actually searched for this space last year, I guess it was, last summer. Right?
Yeah, I remember taking the train in and looking at some spaces.
And so Daniel is here today because he's spending some time at Columbia, and we decided to record right here at the incubator. What do you think of the incubator, Daniel?
I like it. It's convenient. I started my morning coming in on the train, just like I did when we were looking at space. Spent the morning up at Columbia on the wards. I feel like I'm back in the scrubs world again, being the attending. And then just a quick subway ride, and I was here, and it's nice space. I'm looking forward to this.
It's very nice space. Yeah, we're going to be able to do a lot. But we're here for COVID-19 clinical update number 85. What's new, Daniel?
All right. Well, I don't have my clock that I usually look at, so keep me on schedule. But let me start with my quotation. Good decisions come from experience. Experience comes from making bad decisions. And that's Mark Twain. And as we'll get into this, you'll understand we've got decisions. A lot of people here in the United States have some decisions to make, and that'll be relative to our discussion of boosters. But lots going on. But one of the first things I wanted to put out as far as updates is the living guideline from the WHO on drugs for COVID-19 was put out. And I do think it's important to make that comment. This is on drugs. This is not talking about all the non-pharmaceutical interventions, which still are critical, right? Masking, distancing, ventilation, all the other things we've talked about. But if you go to the WHO website, or you can actually go to the British Medical Journal, so the BMJ has this article, and they actually have a really nice visual to walk people through this. And they break it down into disease severity, but I really feel like there should be more timing. But they go through sort of that early period of time when people are just starting to have maybe fever or cough, headache, feeling crummy, but they're not requiring oxygen. At that point, really, it's very limited in what they recommend, and it's really neutralizing monoclonal antibodies. There's a lot of stuff they recommend against ivermectin, except in a clinical trial, recommend really strongly against hydroxychloroquine. The data's in there, right? We have about maybe a 10% mortality increase. Strong recommendation against lopinavir, ritonavir. That's one of our HIV drugs we tried to repurpose here. It's interesting, and I think this is going to be a little bit, remdesivir, there is a recommendation against in all stages, in the mild initial week and also into the next 2 weeks. Now, the corticosteroids, they're going to recommend not giving in that first week. We've talked about this, maybe a 6-fold increase risk of progression if you give them in that first week, but then they have a recommendation in favor, very strong, during that second week in people with oxygen saturation less than 90%. They're using 90% as a cutoff, signs of pneumonia, signs of respiratory distress, but then also sepsis, septic shock, ARDS. And another, which I'll say is sort of a big one in here, is the IL-6 receptor blockade, right? We've talked about tocilizumab, and they're basically saying in those individuals with an oxygen saturation of less than 90% on room air, and we've talked about those that are given steroids but continue to progress, they have a strong recommendation in favor. We've talked about the experience at the Northwell Health System where you really want to get that in early if they start to progress, at least in that reported experience. If you waited until they were on high-flow nasal cannula, till they're on BiPAP, you've waited too long. It wasn't clear that we had much benefit. So just nice to get an update because I know people are always wanting to have sort of a recap, something visual. And this is, I think, a great thing that can be posted around the world in a lot of these clinics.
So, Daniel, how penetrant will that be? Will it reach every little medical center throughout the world?
A lot of times when I've practiced in these areas, you'll actually see something like this stuck up on the wall. So this will get translated from the WHO to the various ministries. And a lot of the ministries will require, health district, number 3 or whatever, will have to have this on the wall. And I think that can be helpful. I mean, some of the things here, the IL-6 receptor blockers, I'm not seeing that in every one of these smaller centers. There's just a huge cost there. Again, monoclonals, there's going to be certain centers where those are. I think the big impact this will have is sort of what you shouldn't be doing. And hopefully this is going to help get the corticosteroid use timed properly, given to the right patients, not given to the wrong patients. All right. Holidays on the horizon, right? So we have updated CDC guidance. I don't know if anyone's planning on celebrating. I'm looking forward to Halloween, which is a great holiday when it comes to, it's a great holiday. We're going to go with that. And they're giving some safer ways to celebrate the holidays here. And they are acknowledging, holiday traditions are really important. They're important to families. They're really important to children. I should say they're really important to those of us who are older, right? This is really important. It's a mental health thing as well. This is when, multiple generations might gather to celebrate the holidays. What can we do to make this safer, right? Nothing is 100% safe. Don't let perfect stand in the way of what can be a good holiday. So the one thing right up front, what is the best way to minimize the COVID-19 risk? Keep your family and friends safer. As per the CDC holiday guidance, get vaccinated if you're eligible. They then go into a couple of the other things. So what can you do? And again, it's going to be, what is it? If you feel like you're repeating yourself, then you're probably saying it long enough. Protect those not eligible for vaccination, such as young children, by getting yourself and other eligible people around them vaccinated. Masks, wear well-fitting masks over your nose and mouth if you are in public indoor settings, and they say if you are not fully vaccinated, but we'll sort of put that in there. The big thing, and I put this in bold, outdoors is safer than indoors. So this is one of my plugs for why Halloween is such a wonderful holiday. Being out there, going door to door, getting those bags filled with candy. Outdoors is much safer than indoors. What you really want to be doing, as per the CDC guidance, and I agree with this, is avoiding crowded, poorly ventilated spaces. If you're sick, if you're not feeling well, don't host or attend a gathering. And get tested. Get tested if you have symptoms. Get tested if you have any concerns. And we've always talked about get tested if you're going to be around a more vulnerable individual. And we're starting to see better access to those rapid tests here in the U.S. And I'm going to hit on those rapid tests in a sort of a story shared with me as we move forward. All right, children. Children are at risk of COVID. Wearing a mask is less traumatic for a child than being hospitalized. And there's a bunch of trackers that I've talked to people about where we can keep track of what's going on among our younger segment. But let's talk a little bit about some data we have here on vaccines in adolescents. So we are hearing that there was great effectiveness for the Pfizer-BioNTech vaccine in adolescents. And in the MMWR, we had the early release of effectiveness of Pfizer-BioNTech mRNA vaccination against COVID-19 hospitalization among persons aged 12 to 18 years, United States, June through September 2021. So this study used a test negative design in which vaccine performance is assessed by comparing the odds of antecedent vaccination among laboratory confirmed case patients hospitalized with COVID-19 and hospitalized controls without COVID-19. There were 464 patients in the final analysis. So this was 179 case patients, 285 controls. So the nice thing here is we're going to get real world experience, right? We saw from our trials this, near 100% efficacy, but what are we seeing out there in the real world? So the vaccine efficacy against COVID-19 hospitalization, we always want to ask what is that efficacy? Vaccine efficacy against COVID-19 hospitalization was 93% during the period when Delta was the predominant variant. Among the 179 COVID-19 case patients, only 3% were vaccinated, 97% were unvaccinated. So overall, 43% of the case patients were admitted to an ICU. Almost half of these adolescents were admitted to the ICU. 16% actually required life support during hospitalization. This is mechanical ventilation. This is vasoactive infusions. This is ECMO. And then 7% of these critically ill patients died. A point I want to make, all of the case patients admitted to the ICU, all critically ill case patients, and all deaths occurred in the unvaccinated patients. Even though we talk about hospitalization, the hospital experience can be quite different. The average length of stay was 5 days for the unvaccinated, but the vaccinated patients were in and out in 3 days with, as you saw, not ending up in the ICU, not ending up ventilated, not seeing death in that group. So I think this becomes important because there's always been a lot of conversation about why are we vaccinating children? Are we doing this just to protect their parents, just to protect more vulnerable adults? And there is an argument there. I think we've heard how many, like over 100,000 children have now lost their primary caregiver. So that is a problem if a child transmits and they lose their caregiver. But we're also seeing evidence here that vaccinating adolescents can really protect them. We've also heard that the White House has a plan and is getting ready for extension of the EUA for the Pfizer-BioNTech down to children 5 through 11. And I have to say, I like their plan. Once authorized, the White House has vowed it will quickly distribute shots across the country. And where are these shots going to go? They're going to go to 25,000 pediatrician offices, primary care sites, as well as hospitals, pharmacies, and community health clinics. For a lot of parents, they really don't want to just go to one of these mass vaccination sites with a military presence. They may want to go to someone that they trust, someone they can have a conversation with, because for a lot of parents, this is a big decision, and they want a trusted person to have that conversation with. All right. Testing. Never miss an opportunity to test. And just a little reminder on the issue of overdiagnosis with PCR and just how great the rapid tests are for picking up people who are infectious. I think it took us a long time to get here. But I'm going to quote an email from a colleague of mine. Quote, I saw an uptick in COVID over the past week. We had 4 negative rapids followed by a positive PCR. Thankfully, we do not clear to go back to school until the PCR. Now, my response was, remember, the negative rapids probably mean not infectious. I say that with 98% confidence. If you could get CT values on those PCRs, I suspect they'll be greater than 30, and those kids not infectious. I would love to stop picking up kids with just low-level viral RNA with PCR and do a test to stay with only rapids. Even more appropriate to think about this as we get more kids vaccinated. So just my take on that. This is what we're seeing. I say this is the rule in my school district, and I think I talked about it before. Individual who didn't feel well, could have done a rapid, and I would have felt comfortable. And we have several studies showing that if that rapid is negative and that child goes to school, we are not seeing transmission. Having to wait for those PCRs, at some point we're going to have a tide change here because a lot of children are missing educational opportunities, and those rapids, as we've seen, are really excellent, over 98% sensitive at picking up folks that are contagious. All right, active vaccination. Never miss an opportunity to vaccinate. And vaccination is how this pandemic ends. Okay, vaccine, the jabs that keep you from getting sick. I'm going to start off here with a public service announcement. Please get your flu shots. We're already starting to see cases of flu. And remember, schools are open. Many places, the masks are off. So that little reprieve we had last year when schools were closed in many areas, when everyone was wearing masks, I don't know if everyone was really doing that, the flu vaccines significantly reduce the risk for hospitalization and death from flu. I feel like we disparage the flu shots a little, but it may be as high as 90% of flu deaths occurring in the unvaccinated. This is certainly described for influenza-associated pediatric deaths. So, think about this not just for you. Maybe you're, very strong and you're not going to get that sick from the flu, but you can be part of that transmission. So if you can get vaccinated, you're doing something for yourself. And I'm going to suggest it's a patriotic thing to do for our country and for our fellow citizens. All right. Lots new on the booster front. First, the article, immunogenicity of standard and extended dosing intervals of BNT162b2 mRNA vaccine, right? So that's the Pfizer BioNTech. This was published in Cell. It's always hard to read a Cell article. These are dense. But this article builds on the idea that maybe getting a second dose of an mRNA vaccine 3 to 4 weeks after the first dose was perhaps a bit rushed. We did that because we were in a pandemic and we wanted to get up to that level. But perhaps we would get an even better response if we waited, right? And this actually feeds into that narrative that I've been hearing, that this dose we're talking about now is really, that third dose is really a better timed second dose. So in this study, extension of the interval between doses, it was introduced in the UK to accelerate population coverage. The authors here are reporting on a sub-study of 589 individuals, and they report that the neutralizing antibody levels were higher after the extended dosing interval. So here we're doing 6 to 14 weeks instead of just that 3 to 4. And they also looked at CD4 T cells. Now, one of the things I will say is the big impact they saw was an increase in peak neutralizing antibodies and B cells, but we're not really seeing as much of an impact on the T cells. So I think that's an important comment there. This sort of relates to the fact that let's call it the air support, that rate protection against hospitalization and death. There may be a lot more T cell involvement there than just the antibodies. All right, the letter to the editor. Differential kinetics of immune responses elicited by COVID-19 vaccines was published in the New England Journal of Medicine. And here the authors reported on antibody and T cell kinetics. They reported that the BNT162b2 and mRNA 1273, right, so it's the Pfizer and the Moderna vaccines, were characterized by high peak antibody responses that declined sharply by 6 months. These responses declined further by 8 months. Antibody titers in the recipients of the, I'm going to switch over to brand names here, the Moderna, the Spikevax vaccine were generally higher than those of the folks that got the Pfizer BioNTech Comirnaty vaccine. Now, the Ad26-CoV-2S, the J&J vaccine, induced lower initial antibody responses, but these responses were relatively stable over the 8-month follow-up period with minimal to no evidence decline. And I will say, if you actually look at the figures in this paper, when you get out to about 8 months, the Pfizer has dropped below the level of the J&J. The Moderna has dropped to the level of the J&J. But what about T cells? At 8 months, the median CD8 T cell response was 0.016 with the Pfizer, 0.017 with the Moderna, 0.12. So about a log higher, about 10-fold higher with the J&J vaccine. So with all three vaccines, the T cell responses showed broad cross-reactivity against SARS-CoV-2 variants. So we are learning more about these different vaccines and the kinetics of the response. And why are we talking so much about this? Because now on Wednesday, 10/20/2021, so the 20th of October, the FDA expanded the use of booster doses of COVID-19 vaccines in eligible populations. So let's go through what exactly this means. And they do a reminder here. So the agency is amending the EUA for the COVID-19 vaccines to allow for the use of, and we're going to go through this here, a single booster of the Moderna COVID-19 vaccine that may be administered at least 6 months after the completion of the primary series to individuals who are 65 years of age and older, 18 through 64 at high risk of severe COVID-19, 18 through 64 with frequent institutional or occupational exposure to SARS-CoV-2, right? So think of your healthcare workers. The use of a single booster dose of the J&J COVID vaccine may be administered at least 2 months after completion of the single dose primary regimen or single dose shot in individuals 18 years of age and older, and they don't qualify. This is anyone who got a J&J, it's been more than 2 months, you can go ahead and get that second J&J shot if you want, or the use of each of these vaccines as a mix and match booster. So going back to those eligible populations, if you got a J&J and you want to do a boost, if you got 2 shots of an mRNA-based and now you want to boost with, instead of Moderna, a Pfizer, instead of Pfizer, a Moderna, instead of mRNA, maybe you want to do a J&J at that point. So they are basically opening this up and giving people options here. And they do clarify that stuff we talked about before, all those people who've been eligible for that third dose of Pfizer-BioNTech, you can go ahead and get another dose of Pfizer-BioNTech if you choose.
Daniel, does it make an improvement if you, say, got Moderna and then get Pfizer, or Pfizer and then Moderna? Like within the mRNA vaccines, does that matter?
So we don't really know. We talked a little bit last time about those antibody studies where there was a slightly higher level of antibodies if you did 2 Modernas and then a Pfizer, at a slightly higher antibody level if you did 2 Pfizers and then a Moderna, but it was really a slight. So I'm not really sure that there's compelling evidence. Certainly no efficacy evidence. We don't have the T-cell data on that yet. So, if you've been doing Pfizer, and you want to do a third, or I think my mom, I think we talked about this, she got her 2 Pfizers and now she wants to join the Spike Vax so she can get the cool T-shirt. She wants to get that third dose. For Moderna, right, it's going to be half of those first 2 doses. I think that's fine. And some people, interesting enough, some people want J&J because they're starting to buy into this concept of heterologous boosting, right? Certainly a lot of buy-in in people who got their first J&J wanting to get 2 shots of the mRNA vaccine.
So I think what you said is really important. Efficacy, we don't know what the antibody levels mean. I think to say that mixing and matching is good requires some efficacy data, and we don't have that.
Yeah, I think that's really key. Like we talked about last time I think the J&J is a perfect example. So you have an individual who got 1 J&J shot, and now they're trying to figure out what to do next, right? So we talked about the phase 3 Ensemble 2 study data where we had efficacy, but this was a very limited number of people. The FDA was a bit critical about how wide those confidence intervals were, but that was at least them trying to present some efficacy if you get a second dose of the J&J. And then some people look at those levels and they say, well, if I have J&J as a background, I'm thinking of that as like natural, infection, natural immunity, and then I get 2 mRNAs, we're extrapolating. But yeah, we do not really have all the efficacy data that we will get, and we will get this. So I like to think that people should have a conversation with a trusted health professional to help them make the choice that is best for them. But I think that actually, I say that, and it makes me think of my friend Lucy McBride, who has this concept, which I agree with, that everyone should have a trusted health professional, a primary care physician that they can reach out to, maybe even by phone for a conversation like this, maybe a video visit, maybe even getting in face-to-face, depending on preference, because these are decisions that you want to feel like you've made the right choice for you. And I've had this conversation. Let's say you're a woman who's pregnant, And you may be thinking, I want to do a certain vaccination, get really high antibody levels so I can transfer them to my unborn child. And I want them to be in the breast milk. But then again, as this has come up, I think, on one of the immunes, there may be more than antibodies that we transfer. So maybe you want a really robust cellular response. Maybe there's actually transfer of cellular protection to the newborn. So a lot of this, you really want to feel like you're making the right choice. And in a perfect context, instead of giving people lottery tickets to get vaccinated, it would be ideal if we gave them the ability to have that conversation and find out what the best choice is for them. What do you think, Vincent? Am I being too idealistic?
Well, I think if they talk to you, they could make a good decision, but I'm not sure that most other primary care physicians are going to be well-versed. They're going to just follow the CDC recommendations, I think.
Yeah. Hopefully we'll get them to listen to this TWiV. But no, it's a challenging. You want to make sure you make the right decision for you. And if you have questions, I guess they can email us here, right?
Daniel@microbe.tv.
All right. So the viral symptom phase, the period of detectable viral replication, what we like to say is the time for monitoring and monoclonals. It's not the time for antibiotics. It's not the time for steroids. And a little more information on monoclonal antibodies. And this was an article, Administration of Monoclonal Antibody for COVID-19 in Patient Homes. This was published as a peer-reviewed research letter in JAMA Network Open. In this publication, the authors describe a program in Michigan at multiple hospitals within the St. Joseph Mercy Health System, where they were providing home MAB infusion to prevent high-risk patients from requiring ED care and hospitalization. And I have to say, this is a topic I'm very excited about. I should give a lot of credit to Ben Wigand, who I worked with at United Health Group, who was repeatedly talking with the FDA, expanding that EUA so that this could be done. Because remember, MABs had to be done initially through the remdesivir network. The EUA was expanded to allow this to happen. And here, among the patients administered MABs in the home, 5.6% were hospitalized following home infusion because of the worsening of the symptoms. But of those folks, the median length of stay was 3.3 days. None required intubation. All were successfully discharged. So I think just gives us a little bit of granularity. Not only did the monoclonals keep people out of the hospital, not only do they protect people from dying, but should they progress and end up in the hospital, it's a much different experience, right? 3.3 days. This is sort of similar to what we were seeing in vaccinated adolescents. And remember, these are high-risk patients. All right. So the early inflammatory phase, right? This is often when people get hospitalized, when they have this cytokine storm or the pulmonary phase. And we haven't talked about colchicine in an article, but what's going on with colchicine? The article, Colchicine in Patients Admitted to Hospital with COVID-19 Recovery, a randomized controlled open label platform trial, was published in The Lancet Respiratory Medicine. And these are the results of the Recovery Collaborative Group. I quote my wife, "What would we do without the UK giving us such informative results?" I mean, I have to say, the Recovery Collaborative Group has really stepped up. Maybe we can move back into the UK's basement and, start to learn how to do, trials like this. But in this study, 5,610, 49% of patients were randomly assigned to the colchicine group, 5,730, so 51% to the usual care group. And what did they find? So overall, 21% of patients in the colchicine group, 21% of patients in the usual care group died within 28 days, so no difference as far as mortality. And I'm basically going to go through median time to discharged alive from the hospital within 28 days, requiring mechanical ventilation at baseline, going on to mechanical ventilation, basically all the way across. No benefit on any parameter to using colchicine, but the usual side effects with colchicine. So I think it's really important here to pause and say that negative data is critical. We need to know what works. We also need to know what doesn't work, because a lot of anecdotes, a lot of stories, a lot of passion about a particular therapy can develop And then we really need proper science to let us know. Otherwise, we leave this vacuum, and I think we've seen how disastrous that vacuum is. But here we are looking at colchicine, really great study, not seeing any benefit here. All right. Another one, and this is one looking at the efficacy of interferon beta 1A plus remdesivir compared with remdesivir alone in hospitalized adults with COVID-19, a double-blind randomized placebo-controlled phase 3 trial. Now here, this is also in The Lancet Respiratory. These are the results of the double-blinded randomized placebo-controlled trial at 63 hospitals across 5 countries. Eligible patients had to be hospitalized, a confirmed PCR test. They had to meet one of the criteria suggesting that lower respiratory tract infections, so radiographic infiltrates, a decreased oxygen saturation. They randomly assign them to the 2 groups. So they're either going to get remdesivir or remdesivir plus the interferon beta. And the interferon beta group in no way was superior to the outcomes of the remdesivir alone. So negative data here for interferon beta compared to the usual standard of care. All right. Well, I want to close here by a couple things, making a couple comments. One is to say no one is safe until everyone is safe. I think it's really important that we continue to look out at the world and see what's happening globally. And we are here getting to the end of October. And August, September, October, all donations made to PWB will support floating doctors. And I want to do a little shout out here to Robert Trotman, who committed to making a generous donation to ensure we meet our goal. Dr. Trotman is an infectious disease specialist in Missouri, and he just won the Humanitarian of the Year Award from the Community Foundation of the Ozarks for all the COVID work he has done. And he's actually going to gift that award so that we can match it and help support floating doctors.
Time for some email questions for Daniel. You can send yours to daniel@microbe.tv. Rachel writes, can an antibody test indicate if a booster is needed? What metric and what level would indicate if a booster would help?
That is an excellent question because I get it all the time. Actually, I have to say when I was at Columbia this morning, I was getting a text, and this was actually from a physician saying, I'm in my 70s. I just checked my antibodies based upon this level. Should I get a booster or not? And I think we need to keep reinforcing we do not know what to make of with these antibody tests. We certainly see people with high antibodies get admitted to the hospital. These might be high antibodies from infection. There might be high antibodies from vaccination. If we keep repeating it over and over again, it does not make it true. We do not know that certain antibody levels are correlates of immunity. So I do not recommend people getting serology tests prior to getting a boost if they're going to get a boost. And this is challenged because we really could use those correlates of immunity. It would be wonderful if I could do a blood test and then be able to tell my patients, you are someone who would benefit from a vaccine. You're someone who would not benefit from that vaccine booster. But no, there's no antibody level below or above that we know can help us make this decision.
Holly writes, first, I love your weekly podcast on TWiV. 20 months ago, I'd never heard of TWiV. Now I am a total groupie. As a nurse practitioner working in a busy primary care practice in the greater Chicago area, your weekly updates have greatly helped my ability to provide scientific and evidence-based medicine to frightened and concerned patients. Thank you. Today, I ordered a PCR COVID test on an older man that I suspect has SARS-CoV-2. I mentioned to him that if he tested positive, I would likely recommend a monoclonal antibody. He was all over it. He's not vaccinated. He then asked me what he could expect his long-term immunity to SARS-CoV-2 would be after receiving a monoclonal. Would it provide protection? If so, for how long? We certainly know that immediate outcomes are better for patients infected after they receive a monoclonal treatment. However, are you able to comment on immunity after monoclonal treatment for COVID?
Yeah, no, so this is a great question. When people go ahead and get the monoclonals, based upon the different monoclonals, and I'm going to use the Regeneron, the Eli Lilly cocktails here, the half-life of these products is in 24, 26 days, sort of right there, a little less than a month. So if you're going to use them, for a recurrent prophylaxis, right, you have to give them every month. So we're thinking 1 month, you get to 2 months, you're at a quarter. By 3 months, it's pretty much down there at about 1/8 of that original dose. So we think that the people then become susceptible to reinfection. So at that point, we're recommending that people get vaccinated. But to go into depth, what about the potential protection that they may have gotten? And actually, I have to admit, a lot of people have said, I don't want monoclonals because I want to get that natural immunity, now that I've gone ahead and gotten COVID. We do not really know the long-term protection that you're going to get. We think that the sooner you get those monoclonals, the shorter period of time that your immune system is going to be exposed to and being educated by this experience. But it's really it's like getting educated, in a war zone, right? You do not want to get educated in a war zone. So we are not sure how much protection, how much immunity you develop. The sooner you get those monoclonal antibodies, the better for outcomes, but also the worse for you developing any immunological memory. Across the board, we say, I understand you're not vaccinated now, but when you hit that 3 months, we really want you to get vaccinated.
Sounds like a good study to do would be to take patients who have been treated with monoclonals, wait till they diminish, and then check their serology to see if they've made their own antibody response, and if so, what level.
And it's not a hard study to do because we could look at nucleocapsid, for instance, which is not going to be in the monoclonals. Yep. You could even, modify your assay so that it's not going to bind, right? Because we know, like, for instance, with the Eli Lilly, you can get around that. But yeah, the nucleocapsid would be easier. But no, this would be interesting. I think people would want to know the answer to this because as much as we encourage people to get vaccinated, there are certain people that are very resistant. Maybe this information would help them make that decision.
Marios writes, a friend of mine has gone on a cycling tour. All the cyclists were fully vaccinated, but the organizers decided that every cyclist should have a PCR test prior to participation. I felt this was a bit over the top. Should an asymptomatic, fully vaccinated person ever have to go for a PCR test to do anything, like fly, etc.? I wanted to know what TWiV thought about this. Maybe a rapid antigen test might be better in this situation.
So I like this. You're thinking this through. So what do we know? We do know that people who've been vaccinated can get infected. We do know that people who have been vaccinated can get infected and can transmit, right? Because we have studies showing a reduction in that, but it's not a reduction to zero. We certainly have case contact series showing that can happen. We do think that risk of infection is significantly lower, at least for a period of time. We do think that risk of transmitting to others is significantly reduced, but it's still there. Now, the antigen tests I love, as you bring up, antigen tests really tell us if this person is contagious, if this person is capable of transmission, right? So do you really want a PCR maybe done within this 72-hour window, or do you want that antigen test right before you get in that plane, right before you go on this bike course? I actually think there's a lot to be said for the antigen test. You get an answer right then. It tells you what's going on right now. But yeah, there are reasons to test people who are vaccinated.
Bill writes, I have a 40-year-old patient who had COVID last year and has long COVID. Should she get the vaccine?
I'm going to say yes, and let me talk a little bit about this. There was a recent TWiV. We had David Tuller on. He's a great guy. He and I have chatted several times. And there came up this question, is there evidence that the vaccines impact in a positive way long COVID? Or is it just anecdotes, what we're sort of seeing? There are 2 studies now. There was the original Bristol study. It was sort of smaller. And then a couple TWiVs back, I talked about the French study. There's no placebo group, so we don't know how much of this is a placebo effect. But what we're seeing, what the French study was demonstrating, if people get vaccinated, they are twice as likely to report complete resolution of symptoms. We are seeing in that study, with all its flaws, that people who got vaccinated were more likely to report improvement in a number of scores. So there is evidence to support a therapeutic impact of vaccination. But there's also the other. If a person has long COVID, they had COVID. They're at risk of reinfection. We know that getting vaccinated will drop that risk of reinfection by more than half.
And finally, Lori writes, I wrote in the summer, I'm a Canadian who works in Kazakhstan, was vaccinated with 2 doses of Sputnik in April, May, went home to Toronto, and I asked you whether it was bad for the body to add another 2 doses of an mRNA vaccine. I got the 2 doses of Pfizer before we returned to Kazakhstan in August. We were meant to have a holiday next week, but my PCR test today came up positive. I'm stunned and honestly a bit freaked out. I teach online. I'm careful to wear masks, but I did have a couple of dentist appointments last week. I don't think dentists here are as careful as they are in Canada. I did some quick reading after I got the result and was further dispirited to find that people with breakthrough infections are often older with underlying conditions. I'm 53, maybe that's older, but healthy. I don't have symptoms yet, but wonder how true those findings are. Are breakthroughs usually in people with weakened immune systems? Is Delta messing with these initial stats?
All right. Well, first I want to start by reassuring that 53 is very young. But here's, and I think we keep returning to this, is vaccines historically have never really been touted as preventing you from getting an infection. They've always been designed to protect you from getting sick, to protect you from hospitalization, death, paralysis. So it was actually a bit of a surprise at how effective these vaccines were shown to be at preventing people from even getting a positive test. But I want to put this in context. The more and more people that get vaccinated, the more often we will see positive tests. I was on a call this week with our urgent care doctors, and one of the urgent care doctors was quite upset. I'm seeing all these positive tests. Yeah, we are seeing positive tests in people who've been vaccinated. They tend to have a much milder course than you would expect compared to someone who is unvaccinated. If you think about, their age, their comorbidities, et cetera. Certainly, people who've been vaccinated have died, but I like to put this in context. If you look at the number of people unvaccinated deaths in the United States, it's over 700,000. If you look at the number of vaccinated individuals that have died of COVID, it's maybe in the 7,000 range. So we had that really tragic loss this week of Colin Powell. And that was, again, an individual who had an immune issue, a cancer of his immune cells that prevented him from getting that protection. At 53, with what you're telling us, I really expect that the vaccines in your case have defanged the SARS-CoV-2, have defanged COVID-19. I expect that for you, this will be, in most likelihood, just the sniffles, a mild cold. In a sense, this will be, I guess, your 5th boost, and you should be good going forward.
That's COVID-19 Clinical Update number 85 with Dr. Daniel Griffin. Thank you, Daniel.
Oh, thank you, and everyone be safe.